Have you ever wondered if your body is still producing spike proteins long after receiving the mRNA COVID-19 vaccine? To understand this question, we first need to understand what mRNA vaccines are and how they work. These vaccines, like Pfizer's and Moderna's COVID-19 shots, introduce a tiny snippet of genetic material called messenger RNA into our bodies. This mRNA instructs our muscle cells to produce copies of the spike protein, a part of the SARS-CoV-2 virus. Once these spike proteins are created, our immune system recognizes them as intruders and quickly mounts a defense, preparing us to fight off the actual virus if we ever come into contact with it. But there have been some rumors and concerns floating around. Some people worry that these spike proteins could continue to be produced in our bodies for longer than expected, potentially leading to health complications. So, is there any truth in these claims? Let's delve into the science and find out. In the world of mRNA vaccines, spike proteins are the stars of the show. They're the ones that put our immune system through its paces, training it to recognize and combat the virus should it ever show up uninvited. But where do these spike proteins come from, and how long do they stick around? Let's take a step back and look at the bigger picture. The Pfizer and Moderna vaccines work by introducing a little piece of genetic material known as messenger RNA or mRNA into our muscle cell. It is like a blueprint instructing our cells to produce copies of the virus's spike protein. It's a clever strategy, isn't it? We're essentially using the virus's own tactics against it, but here's the key. This mRNA shouldn't stick around for long. It should be quickly degraded within a few days. As for the spike proteins themselves, they are not permanent residents either. According to the Infectious Disease Society of America, these spike proteins typically last up to a few weeks in the body. During this time, our immune system is hard at work identifying these foreign proteins and swiftly destroying them. So the spike proteins should not overstay their welcome, but can we test if they are still around? What if you could test whether your body is still producing spike proteins or if they're still in your system? It sounds like a plot from a science fiction novel, but it's actually based on research and medical advancements. There was a study conducted on post-vaccine myocarditis, a heart condition that occasionally arises after vaccination. This study found that individuals with myocarditis had remarkably high levels of full-length spike protein in their plasma. On the flip side, asymptomatic unvaccinated individuals had no free spike protein detected, indicating that the body had successfully eliminated these proteins. But you might be wondering, how do we even test for these spike proteins? Well, there are tests available that can detect the presence of spike protein antibodies in your system. For instance, a spike protein antibody test can provide results within a reference range where higher levels indicate a positive result for anti-SARS-CoV-2. This suggests that your immune system has responded to either the COVID-19 vaccine or a prior infection. However, it's crucial to note that these tests cannot differentiate between antibodies produced in response to the vaccine and those resulting from a previous infection. So, while they can provide useful insights into whether your body has developed an immune response, they can't specifically determine if the body is still producing spike proteins. While these tests provide useful information, they can't specifically determine if the body is still producing spike proteins. So, we're still somewhat in the dark when it comes to understanding the full picture of how our bodies react to these vaccines over the long term. But as science continues to advance, we can hope for more precise answers in the future. So, where does this leave us in our quest to understand if our bodies continue to produce spike proteins long after the mRNA vaccine? Our journey through the intriguing world of mRNA vaccines has led us to some enlightening findings. Let's revisit what we've learned. We discovered that the Pfizer and Moderna mRNA COVID-19 vaccines introduce messenger RNA into our muscle cells. These cells then get busy creating copies of the spike protein. However, this should be a short-lived process. The mRNA degrades within a few days, and the spike proteins typically don't outstay their welcome, usually lasting just a few weeks. We also explored the world of testing for spike proteins. While tests exist to detect spike protein antibodies, they can't tell us if the body is still manufacturing these proteins or if they're just hanging around past their due date. In the context of myocarditis and turbocancers, studies have been conducted, revealing intriguing findings. For instance, full-length, unbound spike proteins were found in the plasma of patients hospitalized with myocarditis after receiving mRNA COVID-19 vaccines, but not in asymptomatic, unvaccinated individuals. In a nutshell, while there's a lot we're still learning about mRNA vaccines, current science indicates that spike proteins should not linger in our bodies for long after vaccination.
Can you test for spike protein production post-mRNA vaccination? This question, quite intriguing and somewhat complex, is one that has been posed by many, and today we aim to delve into it. Firstly, let's unpack the concerns regarding spike protein. These microscopic proteins have been at the center of discussions primarily due to their potential biological actions. Myocarditis, turbo cancers, and other health issues have been speculated to be associated with these spike proteins. So the question arises, can you test for the spike protein? The answer is indeed yes. Scientists employ what is known as a proteomic approach to detect the spike protein. This involves the use of mass spectrometry examination of biological samples to identify specific fragments of the recombinant spike protein in individuals who have received mRNA-based vaccines. Quite fascinating, isn't it? However, the presence of these proteins isn't universal. Studies have found the specific PP spike fragment in half of the biological samples analyzed. Interestingly, the detection of these proteins didn't depend on the SARS-CoV-2 IgG antibody titer. They were found in individuals anywhere between 69 and 187 days post-vaccination. Now let's move to the more concerning topic of turbo cancers. As of now, there are no specific studies on the detection of spike proteins in cases of turbo cancer post-mRNA COVID vaccine. This could be due to a lack of scientific consensus or substantial evidence supporting a link between the two. However, there is an association being studied between cytokines and a hypercatecholaminergic state that could potentially lead to turbo cancers. Cytokines, small proteins essential in controlling the growth and activity of other immune system cells and blood cells, play a crucial role. When released, they signal the immune system to do its job. However, in a hypercatecholaminergic state, such as in severe cases of COVID-19, patients may experience a cytokine storm. This excessive production of cytokines can lead to a hyperinflammatory state, potentially causing acute respiratory distress, pulmonary edema, and multi-organ failure. In summary, the detection of spike proteins post-mRNA COVID vaccination is possible and has been primarily studied in the context of myocarditis. Various methods such as antibody profiling, T-cell analysis, cytokine profiling, and ultrasensitive antigen assays have been used. Proteomic approaches using mass spectrometry have also been employed to detect spike protein fragments. However, there remains a gap in research when it comes to the potential link between spike proteins and turbo cancers. As we continue to navigate the world of medical innovation and discovery, it is essential to stay informed and updated. The quest for knowledge is never-ending, and every step brings us closer to understanding the mysteries of the human body and the viruses that affect it. Do you know what a hypercatecholaminergic state is, or how one can test for it? A hypercatecholaminergic state refers to a condition in which there are elevated levels of catecholamines such as adrenaline, noradrenaline, and dopamine in the body. Testing for the condition is a multi-step process that begins with a thorough clinical assessment by a healthcare professional. Symptoms such as high blood pressure, rapid heart rate, excessive sweating, and anxiety are evaluated. The next step involves blood tests that measure the levels of catecholamines and their metabolites. It's important to note that these tests often require specific conditions like fasting or lying down for a specific period before the test because diet, posture, and stress can affect catecholamine levels. In addition to blood tests, a 24-hour urine test is also utilized. This test measures the amounts of catecholamines and their metabolites excreted over a day. It's a useful tool in determining if there is a persistent elevation in catecholamine levels. When a pheochromocytoma, a type of tumor that can cause excessive catecholamine production, is suspected, imaging studies such as CT scans or MRIs are used. In some cases, genetic testing might be recommended, especially if there's a family history of conditions causing hypercatecholaminergic states. Depending on the suspected underlying cause, additional tests might be necessary. Now, shifting gears to another topic, testing for the presence of spike proteins after COVID-19 mRNA vaccination. Several methods have been employed for this. In a study of myocarditis following mRNA vaccination, full-length spike protein was detected in the plasma of patients using methods like antibody profiling and T-cell analysis. The spike protein was detectable for up to 3 weeks post-vaccination in these cases. There's also the spike protein antibody test. This test measures the presence of antibodies against the spike protein, indicating an immune response to either the vaccine or a previous COVID-19 infection. A proteomic approach using mass spectrometry was employed to detect specific fragments of recombinant spike protein in vaccinated individuals. This method found the PP spike fragment in biological samples up to 187 days after vaccination. Additionally, ultra-sensitive single-molecule array antigen assays were used to detect levels of spike protein and cleaved spike protein in free form or bound to antibodies. In conclusion, the detection of spike proteins post-mRNA COVID-19 vaccination is possible through various methods like plasma analysis, antibody tests, proteomic approaches, and ultrasensitive antigen assays. These tests can detect the presence of spike proteins or related antibodies, indicating an immune response to the vaccine or a previous infection. However, these methods have limitations, such as not being able to specify if the body is still producing spike proteins or if they are still present in the system. That's all for today. Stay curious, stay informed, and never stop learning.